Welcome back to part 2 of the lecture series dealing with modification of basic principles, or modification of Mendelian outcomes, or modification of phenotypic outcomes. The basic premise that we established in the last lecture dealt with these modifications in phenotypic outcome. These are modifications that are different from the classic dominant-recessive relationship that we talked extensively about with Mendel's work. Now, what I also hopefully was able to get across to you in the last lecture, that the behavior of the genes, that is the segregation pattern that Mendel had proposed that occurs, still holds true. And because those principles still hold true, we can still make predictions about the outcome of a specific cross if we know the genetic information of the parents involved in the cross. So again, what we are dealing with in this lecture series are modifications in phenotypic outcome due to specific scenarios, and we did discuss a handful of them in the last lecture. The concept we left off with in the last lecture specifically was known as epistasis. Now, epistasis is an interaction of gene products that occurs in a way that one gene product negatively affects the expression of the other gene product. And again, we had several examples of that in the previous lecture. Today's lecture is going to start off from that same point, dealing with gene product interactions. Now, in this particular case, this is a different type of example because we're not talking about one gene product negatively affecting the expression of the other. It's just simply when gene products interact, oftentimes there are situations that arise where phenotypes are going to turn out different than what we would typically expect, again, in a classic dominant-recessive relationship. And that example is going to be one that you see with this particular slide. With this particular slide, we're going to be dealing with the color of peppers, bell peppers in particular. And the pepper itself is the fruit of the plant. And again, we're dealing with color of that particular fruit. Now, in this example, we're going to be dealing with two different genes. Gene 1 is going to be involved in the color of the pepper. That is, the pigment that is produced that results in the pepper color. This is a two-allele system. It is a classic dominant-recessive relationship. And in this particular case, the two alleles represent red peppers and yellow peppers. Now, in this example, I've deviated a little bit from the norm or the convention of labeling systems because what we discussed again at the beginning of the last lecture was that the convention in using a gene symbol or designation is to use the first letter of the recessive trait. However, in this case, I'm going to reserve Y for the second gene. So for simplicity's sake, to be able to differentiate the two genes, in this case, again, the gene we're interested in, the first gene, is for the color of the pepper, two alleles, red, yellow, classic dominant-recessive relationship, red being dominant to yellow. The second gene in this gene product interaction example is a gene that's involved in chlorophyll production. Now, this is specifically chlorophyll production in the pepper. It is a dominant-recessive relationship that is a classic dominant-recessive relationship. And in this case, we're going to use the letter designation Y. The dominant is actually to not produce chlorophyll in the pepper. And that's just indicated in my handwritten notes there as the uppercase Y being equivalent to no CHL, which is my abbreviation for chlorophyll. The second allele is the recessive, and the recessive is to allow for chlorophyll production in the pepper. Again, that's in the fruit itself. This is different than chlorophyll production in the plant overall. Again, this is specifically in the pepper. Now, when we talk about chlorophyll production, sort of a side note is that chlorophyll has a green color. And it's this green color that is the result of chlorophyll production that is going to play a role in some modifications of phenotypic outcome. All right, so what you have here is a whole series of different scenarios, and I'm going to talk you through what's going on in this example. In the first column, what you're looking at is a modified genotype. It's modified because not the entire genotype is not listed. Only a portion of the genotype is listed. So for example, the very first one is designated with the dominant R for red allele, and then a dash or a space, and in this case, this is actually representing any second allele. So in a situation where the second allele was the dominant, or a situation where the second allele was the recessive, would result in red color being produced, because red is dominant over yellow. And the same holds true for the second gene. For the second gene, I've got the capital letter Y indicating the dominant allele being present, and then a space indicating that the second allele in this genotype can either be dominant or could be recessive. In both of those genotypic scenarios, we would expect for no chlorophyll production in the pepper. So the second sort of column that you see here indicates what color the pepper is, and the far right column indicates the outcome for the specific genotype listed. So let's actually start with that. In a situation where you have one copy of the dominant allele for red pigmentation, doesn't matter what the second copy is, either homozygous red or heterozygous, those individuals would have red pigmentation being produced. For the second gene, where you have a dominant allele for chlorophyll production, the second allele, doesn't matter what that is. This could be a homozygous dominant situation or a heterozygous situation, because no chlorophyll production is dominant to chlorophyll production in the pepper. There's no chlorophyll being produced. So then back to the middle column, what that means is genotypically we have red pigmentation being produced, no chlorophyll being produced, and so overall the phenotype of the pepper is red. Let's move down to the second example. The second example keeps the genetic combination for gene number 2 for chlorophyll production the same, having one copy of the dominant. Doesn't matter what the second copy is, could be homozygous dominant or heterozygous. That would result in no chlorophyll production, indicated sort of in the right column again. However, if we take the example of being homozygous recessive for gene number 1, these plants are going to produce yellow pigment. So these plants will have yellow pigment being produced and no chlorophyll being produced, hence their phenotypic color is yellow. Now I'm going to move down to the last row. In the last row, the genotype that you have here is specific. If you have an individual pepper that is homozygous recessive for color, they would produce yellow pigment. And if the same plant was homozygous recessive for chlorophyll production, they would produce chlorophyll in the pepper. This means yellow pigment is being produced and chlorophyll is being produced in this specific genetic combination. What that means, though, or results in, is a green pepper overall. Even though the yellow pigment is being produced, the green color from the chlorophyll sort of dominates over, I shouldn't use the term dominate, I should use the better term, basically masks the yellow pigment. So even though the yellow pigment is there, because chlorophyll, the green is so intense, that's actually what we see in the pepper color. So let's move back to the row that I omitted. In a situation where you have an individual pepper that is one copy of the dominant allele for color, and doesn't matter what the second copy is, could be homozygous dominant or heterozygous, would produce red pigment. But for the second gene, if that individual was homozygous recessive, that individual plant would produce chlorophyll. So now you have red pigment being produced and chlorophyll being produced, which again has the green color to it. What happens in the overall appearance of the pepper is that it looks sort of brown or muddy in color because now you have the red pigment and the green from the chlorophyll coming together in what turns out to look like a brown pepper. Now again, this is distinct from epistasis because epistasis is where one gene product will have a negative effect on the expression of the second. This example again is purely just when gene products are produced and there is some type of interaction that occurs, you might get a modified outcome in phenotype, like the green and the brown that we just talked about for these peppers. The next concept that might lead to a modification in phenotypic outcome is a concept referred to as pleiotropy. Pleiotropy is a situation where you have the impact of a single gene that basically affects more than one character overall. So, single gene, multiple phenotypic effects. The best example of pleiotropy is the one that you see in this particular slide. In this case, we are looking at an individual that is afflicted with sickle cell anemia. So sickle cell anemia, if you look at the very center of the slide, is a situation where an individual's red blood cells have a sickle shape, basically a crescent shape associated with them. And this is because there is a mutation in the hemoglobin gene that causes the hemoglobin protein to misfold and results in these misshapen red blood cells. Now, individuals who have two copies of the allele that causes this sickled shape, is one gene, so one gene is involved in this example. The initial phenotype is the difference in the shape of the red blood cell. However, the shape of the red blood cell having the sickle shape or the crescent shape actually leads to a whole host of other problems in the individual afflicted with this particular disease. So it results in the breakdown of red blood cells, it results in clumping of cells and clogging of small blood vessels, and it causes accumulation of the sickled cells in the spleen. All right, so that's physiologically what is happening. But then what does that do as we continue sort of looking down the flow chart on this slide? It causes physical weakness, anemia, heart failure, pain and fever, brain damage, damage to other organs, and spleen damage, which can then also lead to impaired mental function, in some cases paralysis, prevalence or predominance of pneumonia and other types of infections, rheumatism, and kidney failure. So even though we have a single gene, there are multiple phenotypic effects that we can see due to that single gene and the mutation associated with that single gene. And this is a classic example of pleiotropy. The next example is polygenic inheritance. Now, in this particular example, it's almost the opposite of what we just talked about with pleiotropy. In this case, you have multiple genes that actually contribute to a single trait. The example we're going to look at here in just a minute is skin color, but eye color is another great example in humans of polygenic inheritance. It's not a single gene that determines the color of eyes in humans. There are multiple genes that are involved. Hence, this is referred to as a polygenic trait or polygenic inheritance. Again, you have multiple genes that are playing a role in a single trait or single phenotypic outcome. Now, eye color, like I mentioned in the previous slide, is a classic example of polygenic trait or polygenic inheritance. But skin color is also another really good example, and that's what we're looking at on this particular slide. In this case, we have multiple genes that are involved in skin pigmentation. So what we're looking at are 3 sets of genes, but in this case it's also the alleles that are present that play a role in the phenotypic outcome. So let's look at the parental generation in this case. We're going to have, in the parental generation, an individual who is true breeding for the 3 genes in question, having the very light alleles associated with them, or light alleles associated with them. So even though it's 3 sets of genes, there are 6 alleles that are present, and in this case, all 6 alleles are instructions for light pigmentation. The opposite extreme of that would be parent number 2, and parent number 2, again, has 3 sets of genes involved in skin pigmentation, and in this case, all 6 alleles in the 3 sets of genes are going to be instructions for dark pigmentation to be produced. The F1 is almost always easy to follow because the only genetic contribution that parent number 1 could make is to pass on the light alleles, and the only genetic contribution that parent number 2 can make is to pass on the dark alleles. So an individual in the F1 will still have 3 sets of genes involved in pigmentation. However, 3 of those alleles are light and 3 of those alleles are dark. Now, if we take 2 individuals with the same genotype, heterozygous for all 3 genes, but more importantly again, 3 light alleles and 3 dark alleles, if we cross those 2 individuals, we can look at a Punnett square, this is a modified Punnett square, and make predictions about the outcome of offspring. What you can see from this table is it's pretty complex. However, if you go to any single box represented in the Punnett square, you can make a determination about what color or how intense the pigmentation is going to be of the skin. So in the top, very top left-hand corner would be an offspring that has all 6 alleles light. And in this case, there's a 1 out of 64 chance that that could happen. But if that were the case, that individual's pigmentation would again be very light because all 6 alleles are light. If you work your way to the opposite bottom right-hand corner is the opposite extreme. An individual that has all 6 alleles, dark alleles, would have very dark pigmentation overall. And again, the probability of this is 1 out of 64. However, what you can see is that there's a whole range of phenotypic outcomes for skin pigmentation depending upon the number of light and number of dark alleles that are present. This is a concept that we will come back to at the very end of the semester dealing with quantitative genetics. But the concept here again is that you have multiple genes playing a role in the phenotypic outcome. And in this case, it's even more complex in that what alleles specifically are present will dictate the phenotypic outcome overall. Now the next concept, or the next series of concepts, all deal with the biological sex of an individual. And what we're going to do is we're going to preface that a little bit here. We will, in our next lecture, with our next unit of material, talk about the sex chromosomes and how sex determination occurs in different types of species, and we'll talk about different examples and modes of sex determination. But for now, let's just suffice it to say that we're going to talk about a mammalian example of sex determination and then how that affects modifications in phenotypic outcome. So the quick history behind this is that in the early 1900s, Thomas Hunt Morgan, who was a Drosophila geneticist, isolated chromosomes from Drosophila, and Drosophila, again, is the common fruit fly. And what he did when he isolated the chromosomes was he was able to determine where the homologous pairs were, but very quickly noted that there was a difference in one set of chromosomes when he isolated them from females compared to when he isolated them from males. In females, the 4th set of chromosomes were very easily matched up, And then in males, they were different in that the chromosomes were not exactly the same in size and shape and structure. What this led to was the mechanism of sex determination that, again, we will talk in more detail about in our next unit of material. But for now, what we're going to do is discuss the fact that there are chromosomes that are referred to as sex chromosomes in Drosophila as well as in all mammalian species. The sex chromosomes are involved in the determination of the biological sex of the offspring. All of the other chromosomes are referred to as autosomes. This is something that you are hopefully familiar with and really is just a review. But these chromosomes, the sex chromosomes, are designated X and Y, and they are in fact distinct from one another. In order for an individual to biologically be female, they have 2 copies of the X chromosome, and individuals have 1 copy of X and 1 copy of Y are female. Now the X and the Y, again, are different chromosomes from one another. There is different genetic information found on the X and the Y. And it's this situation or scenario that leads to some modifications in phenotypic outcome. That's because females have 2 copies of X. Their genotypes would follow the same types of patterns we talked about previously, where an individual could be homozygous or heterozygous in genotype, because again, there are 2 copies of the given chromosome. However, males only have 1 copy of X and 1 copy of Y. They cannot be homozygous or heterozygous because, again, both of those terms refer to 2 alleles in a specific combination. Now, for males, they are referred to as being hemizygous for the genes on X and hemizygous for the genes on Y, because they only have 1 copy of X and 1 copy of Y. So the first example of a modification is something referred to as X linkage. This is the transmission and expression of genes located on the X chromosome. The classic example of X linkage is seen with this particular slide, or the figure on this slide, dealing with Drosophila and eye color. In Drosophila, the wild type eye color is red, and there is a mutation that allows for white eyes to be produced. Now, it turns out that this gene for eye color is located on the X chromosome. However, when the genetic work was done, that wasn't really clear where the gene was located. What came across as something as a very interesting phenomenon was crosses that were done, such as cross A and cross B. In cross A, if you have a true breeding red-eyed female crossed with a true breeding white-eyed male, you would get a specific outcome in the F1 where all of the offspring had red eyes. Didn't matter whether they were male or female. And this follows then a classic dominant recessive pattern. However, interestingly, if you switch the genotype with the sex of the individual, so cross B, where you have a female that is true breeding for white eyes, crossed with a male that is true breeding for red eyes, you very quickly see that there's a difference in the outcome of the F1, in that all of the females are going to have red eyes and all of the males are going to have white eyes. Now, the 1/2 here indicates that half of the offspring are female, half of the offspring are male. But in this case, all the females have red eyes and all the males have white eyes, which is very distinct from the outcome that we just discussed in cross A. If you take these individuals from the F1 and cross them and look at the F2, again, you see a very different outcome in phenotype depending upon the sex of the individual. And it was this complex set of genetics that helped Thomas Hunt Morgan go back to coming up with this concept of X linkage. Now, the best way to use gene symbols in problems working with the X and Y chromosome is to actually designate the X and Y chromosome with the letter X or the letter Y, and then the gene involved or in question would be a superscript. So in this case, we might be referring to the white eye mutation, so we would use the letter W. So we could use an X for females with either a superscript uppercase W, indicating the red allele, or an X with a superscript lowercase w for the white allele. The X designation is the same for males, although males only have one X, and the Y designation, well, this particular gene is not located on Y, it's located on X, hence there's no additional second allele in a genotype in males. In humans, there are over or greater than 250 different X-linked traits. One classic example is hemophilia. Another classic example is red-green color blindness. It turns out there's actually two different forms of red-green color blindness. Duchenne's muscular dystrophy, and another disorder known as faulty tooth enamel, but the first three are ones that you've probably heard of more commonly. And these are X-linked traits or disorders, and they all are due to mutations that are found on the X chromosome that can result in those particular traits that we just talked about. Before I talk about the concepts on this particular slide, let me just take one step back and give you the definition of a sex-linked trait. A sex-linked trait deals with the transmission and expression of genes on sex chromosomes. What we looked at in the last example was a subset of that, which is X-linked, specifically dealing with genes located on the X chromosome. But the broader term or definition is sex-linked, dealing with the transmission and expression of genes on a sex chromosome. That is going to actually lead us into the next concept, because we're going to expand. Instead of just talking about X-linked traits, we're going to talk about Y-linked traits as well. But the broader concept is sex-limited inheritance, or a sex-limited trait. This is going to be one that is expressed in one sex or the other, regardless of genotype involved. Now, the first example is a really straightforward example and is the opposite of what we just talked about with X-linked. Now, X-linked traits can show up in both males and females depending upon the genotype of the individual in question. A sex-limited trait is going to be something that is expressed only in one sex or the other. And again, the first example are genes that are referred to as being holandric genes. These are genes that are on the Y chromosome. Only males have the Y chromosome, so only males will have the genes located on the Y chromosome. And in this case, this is a sex-linked trait because it is on the Y chromosome, one of the sex chromosomes, but it is also sex-limited because you will only see the expression of these genes in males because males are the ones that have the Y chromosome and females do not. A little more complex example would be genes that are autosomal genes. Autosomal genes are going to be genes located on the chromosomes that are not sex chromosomes. And one of the classic examples of this is for milk production in mammals. It turns out that there is a gene involved in the level of milk production in mammals, classic dominant recessive relationship on an autosome, not on a sex chromosome. Classic dominant recessive relationship so that we can have a homozygous dominant genotype, we can have a heterozygous genotype, and we can have a homozygous recessive genotype. With those genotypes in females, that will dictate the level of milk production in the female. However, even though these genes are present in the male, it doesn't matter what the genotype is of the male because other circumstances have not allowed for the breast tissue to develop. The hormones are not present to allow for milk production, so it doesn't matter whether the level will be dictated by the genotype as being lots of milk or little milk production if they don't have the biology there to back up the genetic information that is suggested in their genotype. So for males, their genotype doesn't matter because they won't produce milk at all. So this slide really just backs up the concept that we talked about just a minute ago with sex linkage and a sex-linked gene. Any gene located on the sex chromosome is called sex-linked, and the table really nicely highlights for you the differences that will occur in females versus males with the genotypes and what effect that genotype is going to have. The last example I want to leave you dealing with sex chromosomes or the sex of the individual is something referred to as a sex-influence gene or a sex-influence trait. And a sex-influence trait really is just going to be the phenotypic expression is going to be conditioned by the sex of the individual. In this case, you would have expression in both males and females of a given genotype, but the levels of expression may differ between males and females. So this is different from the last example that we just talked about, where you might have expression in one sex versus the other. In this case, you would have some level of expression in both sexes, but again, the level would be different. And classic examples of this are the cleft palate in humans, horns in sheep, pattern baldness in humans as well, are sex-influenced traits. This is all due to the fact that hormonal differences are going to be playing a role in phenotypic outcome. The levels that are being expressed are different in the two sexes. Now, just to summarize those terms, I have them listed here for you. Sex-linked means it's going to be a gene that's located on the X or the Y, the sex chromosomes. Sex-limited means it's limited to one sex or the other, all or none expression by sex. And then sex-influenced is going to be genotype, is going to play a role, genotype and the sex, so genotype plus sex of the individual will determine the phenotype. There will be differences in the levels depending upon if the individual is male or female. Now that concept leads into another concept, and that is phenotype depends upon the environment and the genes. In this case, oftentimes you've heard the term nature versus nurture. Is it your genes or is it your upbringing that cause a specific outcome to take place? And the answer is that it's really complicated. It's not a straightforward answer. The phenotype is going to depend on environmental conditions as well as genetic information present. There are lots of different examples, and we will come back to this concept in a little bit further down in the semester. But this figure that you see here with the two flowers, this is a hydrangea, and in this particular example, you have two different flowers, flower colors, the purplish color on the left and the pink color on the right. In this case, you have a flower that has the exact same genotype for flower color. Well, you look at them and you go, well, they don't look the same. Even though their genotype is exactly the same for flower color, the color is distinct in both of these. And this is because environmental factors do play a role in the phenotypic outcome in this particular case. So again, you might question, well, what's happening here? If the genotype is exactly the same, why are the two flower colors different? It turns out that the pH of the soil that the plant is growing in will have an effect on the pigment production in a specific way to allow for these differences in phenotypic outcome to take place. So in this case, it's a classic example of genotype and environment playing a role in the phenotype. And again, there are many more complex examples of this. Another classic example would be a very extreme example, one that I don't necessarily have a figure for. But if you have an individual who is genetically predisposed to be very tall, so whatever that genetic condition is, to be very tall. If you malnourish that person from the get-go, from birth on, would that individual see their true potential of height just because their genetic information says that they are supposed to be tall? If you're not providing that individual with the proper nutrients, that individual is not going to grow and see that potential. So again, an extreme example, but that the environment does play a role in phenotypic outcome in many situations. Not all situations, but there are plenty of examples where that is true. Now, because I've just introduced this concept that gene expression is often controlled by genotype and the environment, that leads to some additional terminology that we need to define as well as discuss. The first example that I want to talk about is a concept known as penetrance. Penetrance is a quantitative term, and it refers to the frequency with which individuals of a given genotype exhibit some degree of phenotype associated with that trait. The best example of that is to look at the figure on the right, where you have these dots. So in the very top example, what we're looking at is, first of all, we're looking at phenotypic expression with the dots and the colors associated with it, pink or white. Each circle is going to represent an individual. So a situation where there is variable penetrance is a situation where you have the same genotype, but the phenotypic outcome can vary from individual to individual. And again, this is considered a frequency. The frequency with which individuals of a given genotype exhibit that particular phenotype. So that's why we have differences between the pink and the white, okay? The next example is expressivity. Now, expressivity deals with the degree or range in which a phenotype for a given trait is expressed. This is a qualitative term. The degree an allele is expressed when it is expressed. So in the middle example, what you're looking at is variable expressivity. Here you now have variations in the level of expression. So rather than just pink and white like we saw in the top example, you now have variations in the intensity of the pink. You've got very light pink, you've got medium pink, you've got darker pink, and you've got really dark pink. So these are differences in the level of expression overall of a given genotype, resulting in variations in phenotype. The next example on this particular slide has to do with temperature. And temperature does in fact play a role in many situations, and that's because gene products can be sensitive to temperature. The example that you see here is a Siamese cat and a Himalayan rabbit. What you want to notice from both of these images is that the body of the animal is light in color, and the extremities, the paws, the ears, the tips of the nose, are darker in color. And this is due to the fact that the enzyme that is involved in producing the pigmentation works at the lower temperature, at the extremities, whereas the closer you get to the core of the body, the enzyme loses its function and the pigment doesn't get produced. So the core of the body of these animals are lacking in pigmentation, whereas the extremities, where it's colder, is definitely allowing for the enzymes to function. So even though the genetic information is there, the differences in temperature in these individuals is going to play a role in the phenotypic outcome. The last example is referred to as onset of genetic expression. This just means when is something going to be expressed in an individual, and that can vary. So even though a given genotype is present, when the expression of that specific genotype occurs in the lifespan of the individual can definitely vary. This is oftentimes seen with many disorders in humans. So, for example, Tay-Sachs disease is a problem with lipid metabolism. It is autosomal recessive, but in this case, individuals will typically not survive past the age of 3. Another extreme example would be Huntington's disease. Huntington's disease is a brain deterioration disorder. It is autosomal dominant. However, you typically do not see the onset of this particular disease until an individual is in their 30s. So, again, even though the genetic information is present for a given disorder, the time of the onset of a given situation or disorder can vary depending upon, again, a number of factors. So what you've had here with this lecture and the previous lecture, again, are these modifications in phenotypic outcome, and hopefully they've become clearer to you, and what you're getting a good understanding of is that genetics is actually really complex. It's not as straightforward necessarily as we think it is.